I'm Jim Solomon, and I'm the curator of the herbarium at the Missouri Botanical Garden. Since the garden's founding in 1859, our herbarium collection has grown. We are the second largest herbarium in the United States. A herbarium collection is essentially a collection of pressed and dried plant specimens that are mounted on sheets with accompanying information that tells when they were collected, where they were collected, by whom, and oftentimes information about the plant communities in which they grow. Plants lend themselves very well to being mounted on sheets of paper and being organized in folders like these. They can be pressed and then dried, and once they're dried, they are more or less permanently preserved. People have been collecting herbarium material and preserving it in museum collections since the middle of the 16th century. The reason we have these collections is that in order to evaluate and understand and learn about biodiversity, we have to have samples of those species that are out there. So for some species, we may only have 1 or 2 specimens. For others, we may have many hundreds of specimens. Some species are very common, some are quite rare. And so this essentially is the only permanently preserved documentation of these actual samples from living plants that have been converted into specimens that are dried and preserved that we can go back to over and over again to answer basic questions about those organisms. What do they look like? Where do they occur? How might they be related to one another, and so forth. Well, our orchid collection is, I would say, a generalized collection with a very strong emphasis on those geographical areas where we work, such as Central America, Andean South America, tropical Africa, and Madagascar. And many of the species that are represented in the herbarium collection are not necessarily in our living collection. There are many plants that we do not have living. And you can learn a lot of different kinds of things from the living ones in terms of floral morphology and flowering times and so forth. But you can also learn much of the same kind of information from pressed and dried plant specimens in herbarium collections, because they will have dates with flowering. There will be the morphology, the structures that the plants have will be preserved. One of the difficulties of working with herbarium material is the fact that orchids have such interesting three-dimensional zygomorphic flowers that it's very difficult to preserve those in a really good, interpretable way on dried material. That's why oftentimes orchid flowers are also collected in spirits, in other words, in alcohol, to be liquid preserved, because that preserves their three-dimensional structure, even though they will lose all their color in the preservative. Here on this specimen, you can see here a fruit that's developing, and over here there are floral buds and dried flowers. And of course, when all plant materials dry, they shrink and change shape. You can rehydrate some of these flowers to get a better sense of what they looked like when they were alive. but it doesn't ever really return them completely to their original three-dimensional structure, which is why the floral specimens in spirits are really important adjuncts to the pressed and dried plants. The orchid family is the largest plant family in the world. There's something on the order of 27,000 species of named orchids. They occur in all kinds of habitats, from Arctic to moist tropical. They have a variety of different life forms. Some are terrestrial, some are epiphytes, meaning that they grow up on the branches of trees. They're not parasites, but they are simply using the tree as a structural support for getting them up into the canopy where they can receive light. And many of those epiphytic orchids have very interesting roots. This is an example of one specimen of an orchid that has a very large mass of fibrous roots. Oftentimes they're quite pale or white in color. They are surrounded by a special epidermis called a vellum, which is very good at absorbing water. So that means that when you're sitting up there in the canopy of a tree, the only water you ever get is what falls directly on you from rain, or what runs down the trunk or the branches of the trees and goes by. So orchids have to be very, what shall we say, lively, and have developed root systems that will take advantage of any water that comes by and suck it up, and they will store it in their oftentimes somewhat fleshy leaf bases or the leaf tissues themselves. Natural history museum collections like ours are incredibly valuable because they are the only permanently preserved samples of the diversity that exists out there. And if you think about it in terms of how many individual plants are there on the surface of the earth right now at this second, it's an astronomically large number. If you then multiply that by saying, okay, how many individual plants have lived on the surface of the earth since people started collecting specimens in the 16th century, it's an even larger number. And yet there are only about 3,000 museum collections that contain herbarium specimens, and they have about 380 million specimens amongst them. That's a lot of material. But as a percentage of the total diversity that's out there, it is a vanishingly small sample, and yet it is the primary source of our information about the diversity that exists on Earth right now. So I tell people that, this is our natural history patrimony. This is the stuff that tells us about what the biodiversity of the Earth is like. And it doesn't matter whether it's plants or animals. And without this, we would not be able to communicate with each other about that diversity. We have to have names that we can use and talk to each other about particular species. So anytime you ever use a scientific name, you are using a name that is based on material like this, housed in those museum collections around the world. And in order to really understand it, you have to be able to have access to those specimens, and you have to be able to use them and study them to help you learn more about those organisms, not just what their name is. That's my soapbox, Okay.